Hi everyone, welcome back to my channel Pharmazest. Myself Dr. Haimavati. In today's video we will be learning about virtual screening. So we will see what is virtual screening, what are the different types of screening and how the screening process is done. So now let's begin our video. So coming to the definition of virtual screening. So what is actually virtual screening? So it is a computational technique which is used in drug discovery to search libraries of small molecules in order to identify the structures which are more likely to bind to a drug target, that is a protein receptor or enzyme. That means this virtual screening is a computational technique. So with the help of different softwares and computational tools, what we do, we search libraries of small molecules. So there are a number of small molecule databases like PubChem, Zinc, or else there are in-house laboratories. So every pharmaceutical company has its own in-house library of compounds. So there will be millions of compounds. And even small research groups like academic research groups in universities also, they have their own in-house built libraries. So in this they design different ligands and they will store. So what virtual screening actually does is it reduces the number of molecules to a manageable size. That means all the molecules which we design, we cannot synthesize each and every molecule and then test it in the laboratory. So because the compounds are in millions, so if you want to synthesize each and every compound, so it will take a lot of time as well as a lot of efforts are involved and cost is also involved. So in order to reduce this huge process, what we do is we adopt a method that is called virtual screening. So in this we apply different filters so that these millions of compounds, they can be reduced to a manageable size, and these are targeted against a protein which is in question. That means whichever protein receptor you are studying, so it can be a protein target against a cancer disease or a tuberculosis disease, like that. So we take a protein target and then we screen these compounds against that protein. In that way this virtual screening helps us. Okay, so now let's see. So this structure is a protein structure. So you can see it is a cyan blue structure. So cartoon representation of a protein structure, and these red spheres at the center, they are the active site residues of this protein. So in virtual screening, initially what we do is we need to identify a target for any disease. Suppose if you are studying about cancer, so you will identify the proteins which are responsible for causing cancer. And then what you want to do, you want to identify a inhibitor which can inhibit this protein so that you can prevent the cancer disease. So for that what you have to do, first you have to identify a protein target, and after identifying the protein target we identify its active site residues. So which residues are present at the active site, so where the molecule goes and binds to the residues of the protein, that is called the active site of the protein. So first we have to identify the protein and then we have to identify the active site of the protein. After that we take the small molecules and dock it against that. So the first step in this virtual screening process is protein and ligand preparation. So whichever software, you can take any software, it can be Schrodinger, Glide, or it can be Autodoc or any other software. So for all the softwares, the preparation steps are the same. So the first step is preparation of the protein. So in this what we do is, in the protein if there are any unnecessary residues or troublesome residues, they are removed, and apart from that water molecules are also removed and it is energy minimized so that you get a stable structure. After that, the ligands. So we take a molecules, small molecules. All those small molecules are also checked for their structure. So whether they are geometrically optimized or not, stereochemically correct or not, all these factors are checked, and then they are also energy minimized so that you get a stable conformation. And after that, what we do is we generate a grid around the active site of the protein. So as I have explained already, there is an active site for every protein where the molecule will go and bind at the residues which are present in the active site. So at that active site residue, what we do, we generate a grid around the active site so that this grid indicates, so whatever molecule, instead of searching the entire space of the protein structure, it will go and bind only in the grid defined area. So the molecules, they will come and they will bind only in this grid area. And next step is taking the small molecules and applying the different filters. So this is a representation of a filter. So I to show that it is a screening process. So screening is nothing but a filter. So here I have taken two different databases. So these two different databases, they are containing nearly 1,30,000 compounds. So out of this 1,30,000 compounds, we cannot synthesize each and every compound. So we apply virtual screening. In this, what we do, we apply different filters like Lipinski rule of 5, and then reactive groups, if they are present, they are eliminated, and if any bad poses are there, that means if the molecule is not binding to the protein correctly, like then those molecules are also removed. And finally you will get some hundreds of compounds, maybe around 100 to 500, based on the number of filters you have applied. And then we do the binding site analysis. So in this binding site analysis, so the lead molecules which are obtained, we check for the interaction of these ligands with the protein. After that binding site analysis, finally we come to a manageable number of potential leads. So these potential leads, they can be taken up for further study. So you can, another definition which you can give for virtual screening is, it is also called in silico screening. It is a new branch of medicinal chemistry. So this virtual screening or else in silico screening, it is a new branch of medicinal chemistry that represents a fast and cost effective tool for computationally screening compound databases in search of novel drug leads. So it is a branch of chemistry and it is a fast and cost effective tool. So the process becomes faster and the cost is also reduced because you are bringing down the millions of molecules into a manageable number of compounds, so that only the potential leads will be synthesized and tested against the protein. So that is why it is a cost effective tool. Now the basic goal of virtual screening is reducing the number of small organic molecules. So here the reduction of the enormous virtual chemical space of small organic molecules is done. So as I have already explained, so even if all the pharmaceutical companies, they have their own in-house laboratories, the research groups, they have their own in-house laboratories, and we have publicly available databases like PubChem and Zinc, and many other databases are also there, as well as commercial databases are also there. So out of these millions of compounds, we reduce the number of compounds to a manageable number. And then to synthesize or screen against a specific target protein to a manageable number of compounds that exhibit the highest chance to lead to a drug candidate. So the ultimate goal of screening is identifying the compound which has the highest chance to become a drug candidate. And now virtual screening. So how many types of virtual screening are there? So the first one is structure based virtual screening. So if the protein structure is available, we take the protein and we identify the active site and we do the virtual screening. Suppose if the protein structure is not available, Then we do the screening based on ligand based. So this is also called pharmacophore modeling. And we apply different filters like Lipinski rule of 5, reactive groups, bad pose. So Lipinski rule of 5 means if the molecular weight of the compound is more than 500, we eliminate the compounds. So only the compounds which are having a molecular weight less than 500 are only retained, and the remaining compounds are eliminated. So that is one filter. Similarly, there are other filters like number of hydrogen bond donors, hydrogen bond acceptors. So these criteria are also applied. And reactive groups, if any reactive groups are present, they are also eliminated. Suppose if the molecule contains any reactive group, so what happens in the next stages, it will may cause side effects or unwanted reactions. So in order to eliminate those, these reactive groups are also eliminated. And if there are any bad poses, that means if it does not make any favorable interactions with the protein, then also the compound is eliminated from the process. So this is called virtual screening. So here you can see we take different databases and we apply all these filters. After applying the filters, the compounds are reduced to a manageable size, which become the hit molecules and which can be taken up for further study. So as a case study, this is my article. So this is my own research article which I have published in Molecular Diversity Journal. So it is titled as Structure Based Drug Design, Synthesis and Screening of MMA1, that is mycolic acid methyltransferase inhibitors as novel anti-TB agents. So in this what I have done is I identified novel molecules in order to inhibit this protein known as mycolic acid methyltransferase. So it is a target enzyme for tuberculosis. So if you are interested, you can go through this article and I have explained all the steps in there also. I will give the link to the article in the description. So if you are interested, you can go and check the article. And next, after identifying the inhibitors and after performing virtual screening, what we do is we check the interactions. So after doing virtual screening, that is docking process, what happens is we get the glide score and glide energy. So based on this glide score and glide energy, we prioritize all the molecules and we check their hydrogen bond interactions and pi-pi interactions. So these molecules, when they are bound to the protein, they make different types of interactions like hydrogen bond interactions, pi-pi interactions, van der Waal interactions, etc. So based on the interaction, we get the docking score and docking energy. And these are helpful in prioritizing those molecules. And after that, what we do, this is the three-dimensional representation of this docking process. So where you can see this ligand at the center. So this blue color stick representation is the ligand, and this uh, gray color cartoon representation is the protein. So this ligand is making interactions with this protein. So these dashed lines, they are the hydrogen bond interactions, and these orange lines, they are the pi interactions. And here on the right side you can see the two-dimensional diagram of this. So here you can see the ligand is in the 2D representation, and these small circles, the small colored structures, they are the residues of the protein. So here you can see this oxygen molecule is making interactions with this glycine molecule over here, and here this NH, it is making interactions with this aspirin 19, and like that. So we check for all these interactions and we take up these compounds for further screening. So this is about virtual screening. So virtual screening helps in reducing the number of compounds to a manageable size. It is a cost effective and fast tool, and it is a computational technique which helps us to reduce the size of the compounds to a manageable size so that we can take up it for further studies. I hope you understood the concept. If you like the video, do not forget to like, share and subscribe to my channel. See you in the next one.